Hi, everyone. Thank you for joining us. My name is Nico, and I'll be your host for today's webinar, Visualizing Glymphatic Clearance and Peripheral Excretion Routes of Cisterna Magna Infused Tracers with Cryo-Fluorescence Tomography. In this session, we will be covering the use of 3D Cryo-Fluorescence Tomography, or CFT, as a novel neuroimaging technique to study the glymphatic system. Our goal is to give you practical insights, helpful takeaways, and a better understanding of the role of CFT in preclinical research. Before we get started, I'd like to go over a few housekeeping items. This session is being recorded, and we will share the recording with you all after the webinar. Also, if you have any questions during the presentation, please feel free to submit them in the Q&A panel at any time. We will try to address as many as we can during the Q&A session at the end of the webinar. Now it is my pleasure to introduce the speakers. Firstly, we have Dr. Ian Harrison, who is a Principal Research Fellow at University College London, focusing on the glymphatic system and its role in neurodegeneration. His research aims to harness brain clearance pathways as therapeutic strategies in neurodegenerative disease, with the goal of reducing protein aggregation, improving symptoms, and advancing disease-modifying treatments into the clinic. Also joining us today is Dr. Matt Silva, who is the CEO of Emit Imaging, and will be kicking off the webinar with a quick background and overview of CFT. With that, let's get started. Dr. Silva, please take it away.
Thank you very much, Nico. Thank you, everyone, for attending today. Thank you, Dr. Harrison, for joining us. Really looking forward to hearing more about your research. To Nico's point, I just wanted to take a couple of minutes, talk a little bit about what CFT is so that Ian doesn't have to spend time doing that as a part of his presentation. CFT is a whole body ex vivo volumetric imaging tool that we developed about a decade ago, largely because we saw an opportunity to retain whole animal imaging in 3 dimensions, so large tissue volumes, but at much higher resolution than you're typically able to get from standard MRI, CT, or PET imaging. And in that regard, bridge what we were doing in vivo with microscopy tools. So we developed a tool, I'll show you how it works just in one slide, that enables visualization of both anatomical and fluorescence imaging data from samples at relatively high sensitivity, so near to that of nuclear medicine imaging down single-digit nanomolar, and also at high spatial resolution relative to in vivo, so really a mesoscopic zone in between what you see from microscopy and whole animals, so anywhere between 20 to 50 micron pixel sizes. We also, because it's fluorescence and you can do multiple fluorophores or channels at the same time, we designed the system to run from green to near IR. So it covers the full visible spectrum and matches well with existing research that's done today. And interestingly, as you can see on the slide here, Ian may not be aware of this, but one of the early studies that we did, this was probably 2016-ish, was the biodistribution of a Cy5 labeled antisense oligonucleotide administered into the intrathecal space. And so this was tracked by in vivo imaging, but then also we did CFT to show not only the distribution through the CSF, but as you can see here, we can map multiple lymph nodes associated with clearance. So the idea of looking at whole body distribution is something that's always been a value proposition associated with CFT. So how does this technology work? Well, very briefly, we take tissue samples or whole animals. In this case, I'm only showing you one, but we can do multiple at the same time, up to 10. Freeze the sample in a block, and then do serial block face imaging. So as you can see in the animation in the middle, we acquire a bunch of images, depending on the fluorophores, but also anatomical white light images, section, and then repeat. So anywhere between several hundred to a couple of thousand sections later, you have a full 3-dimensional molecular image. A great example of one of the applications as presented recently by Cy from Regeneron, an early adopter of the technology. Unfortunately, we have auto advance on. Let me go back. The team at Regeneron is using CFT to look at the distribution of antibodies delivering viral therapies. So in this case, going left to right in the images, a wild type AAV9 injected intravenous into a mouse results in protein expression throughout the entire animal. By targeting the CNS through the transferrin receptor, so effectively conjugating the AAV9 virus to an anti-transferrin receptor antibody, you can see that there is now much more exquisite uptake in the CNS. However, liver, bone, and a few other areas still show protein expression. By effectively modifying the virus, detargeting liver and some of the other tissues, Cy presented in this work really exquisite targeting of the CNS. And CFT is a great tool to monitor this effect. Lastly, I just want to show that we believe that CFT is a real difference maker, a real inflection point in molecular imaging, but still sits alongside some other really great tools. Here's an example from Ratio Therapeutics showing the biodistribution of a fluorescently labeled small molecule on the left, examined by CFT. That same fluorescent molecule, when imaged by two-dimensional imaging, it can be good to look at time course. However, light in tissue is very complicated. So one might draw some incorrect conclusions just by 2D fluorescence, as you see in the middle, as it would appear that the drug is sitting in the spine and the tumor. But this is just related to the fact that it's a really strong laser, and there's not a lot of tissue between the spine of the mouse and the detector. On the right-hand side, up until this point, really the gold standard that we all would have used would have been radio labeling, which is a highly sensitive technique that can be translated into the clinic, but is radioactive, it's decaying, it's more complicated, and is far more costly. So CFT we find to be not only a viable, but in many cases a superior solution to understand whole body drug distribution and protein expression. So with that sort of just basis of understanding of what CFT is and how it works, would love to turn it over to Ian. Really looking forward to hearing about your research. Take it away, sir.
Thank you very much. So hopefully I can now advance the slides. Perfect. Thank you very much, Matt. And that's a fantastic introduction because it means that I don't have to give an introduction to the technology. So thank you very much for the invitation to speak today. So my name's Ian Harrison. I'm a Principal Research Fellow at the Centre of Advanced Biomedical Imaging at University College London. So I'm going to tell you about some of the work that we've done using this technology, Cryo-Fluorescence Tomography, visualizing the glymphatic system in the brains of animals that we've performed cisterna magna infusion of tracers in. So the glymphatic system, if you haven't come across this pathway before, was first described by Jeff Iliff when he was a postdoc in Mike and Nedgard's group in this paper. And basically it describes a whole brain system for fluid movement. It's thought that the cerebrospinal fluid that kind of surrounds and penetrates the brain travels into the brain parenchyma via a para-arterial influx mechanism. It then exchanges with the interstitial fluid and then is cleared out of the brain via a paravenous efflux mechanism. And it's thought, let me just put a pointer on, and it's thought that the movement of this fluid from the para-arterial space, exchange with the interstitial space, and then clearance via the paravenous space, the movement of fluid in this directional manner takes interstitial solutes with it out of the brain, including proteins that we know are involved in neurodegenerative diseases, which is where my research comes in. So the first experiments used to define this system, what the researchers did was that they infused fluorescent tracers into the cisterna magna of mice, and using techniques such as two-photon microscopy, they were able to show that when you infuse these tracers into the cisterna magna and image the cortical surface via a cranial window approach into photon microscopy, you can see that these CSF-administered tracers travel into the brain parenchyma via this para-arterial influx mechanism. So you can see that the optical layers going down from the cortical surface here, you can see that the green contrast agent travels parallel to blood flow in arteries on the penetrating arteries and arterioles on the cortical surface of the mouse brain. So we got this procedure up and running in our lab, and what it basically involves is infusion, or surgical cannulation of the cisterna magna, which involves fitting a tiny CSF cannula by puncturing the cisterna magna, puncturing the dura, and then advancing a cannula into the cisterna magna, which is just below the cerebellum, you can see here. And what we've been doing in our lab is using this technique to assess glymphatic function in the brain. So rather than taking a fluorescence approach to begin with, like Mike and Nedegaard's group did, what we began to do initially was to infuse MRI-based tracers, so things like gadolinium DTPA. This gives us the specific advantage of, rather than just looking at superficial brain regions, such as a small area of the cortex, we can get an idea of how that gadolinium tracer goes from the CSF into the brain parenchyma in the context of a whole brain. So by using this technique, we infuse gadolinium via the cisterna magna, this dynamic contrast enhanced MRI, and we're able to get an idea of where that tracer goes and how much of it gets into the brain. And we're able to use this technique to show that in an animal model of tauopathy related to Alzheimer's, that this glymphatic pathway in the brain is distinctly impaired. So we've since been using this technique and going back to fluorescent tracers because it gives us a kind of tunable molecular weight signal. So we can infuse cocktails of different molecular weight tracers to get an idea of the size discrimination limits of this glymphatic pathway in the brain. So routinely in our lab, what we do is to kind of infuse two or three different molecular weight tracers at the same time into the cisterna magna, cull the animal, harvest tissue, and then we can do things like imaging the whole brain using IVIS, or we can section the brain and use fluorescence microscopy, or again repeat imaging on the IVIS. And we use this technique in a number of different papers now, showing able to look at the relationship between the activity of this glymphatic system and the accumulation of proteins in the brain. More recently, we've started moving to perfuse fixation and tissue clearing, and using again excising the tissue, so taking out the brain, clearing it, and then using techniques such as light sheet microscopy, we can get an idea of where these different traces go. And you can see this is a ventral aspect, a ventral view of a mouse's brain, and it's been infused with two different molecular weight tracers: a red, smaller cisterna magna dye, and a larger one which kind of retains in the perivascular spaces, as you can see here. So one of the great things about from my point of view, from cryo-fluorescence tomography, is its ability to overcome some of the challenges that we've had imaging the glymphatic system to date. So things like using traditional histology measures, or removing the tissue from the animal, sectioning it. Obviously, we have tissue collection bias. You're kind of dependent on which tissues you take. We can assess the glymphatic system using different biochemical assays. For example, we could inject proteins of interest and look at their clearance in CSF samples, for example. But again, that is quite time consuming, and it doesn't give us the spatial information that we need. And again, other optical techniques such as the ones that I've shown you so far, using IVIS imaging or light sheet imaging, is fantastic. But light sheet imaging, especially, we're kind of limited with the current technologies. We're limited to our field of view, so we can only kind of image the brain once it's excised from the body. A CFT, on the other hand, in terms of looking at glymphatic function, not only in the brain, but looking at glymphatic clearance of whole body clearance of tracers, gives us ex vivo volumetric imaging. The fantastic thing about it is that it can be combined with the anatomical imaging which is acquired at the same time. You can co-register these two imaging modalities to get really accurate segmentation analysis of your regions of interest. It also is really highly sensitive, so we can inject really small volumes or really small doses of these fluorescent tracers to get an idea of where they're going in the brain. So I'm going to tell you about some of the initial work that we've been doing, looking at cryofluorescence tomography and imaging the glymphatic system. So the CFT workflow that we went about is by doing this cisterna magna infusion, as I talked to you about. So we surgically implant a cannula into the cisterna magna, and then in this experiment we infused Texas Red D3, which is a 3 kilodalton Texas Red conjugated tracer, and that's infused in artificial CSF. The animals were then culled at different time points. So here we culled the animals at either 30 minutes, 60 minutes, or 120 minutes post-infusion of that tracer. The animals were then snap frozen and then block embedded, like Matt already mentioned. In our experiment, we were able to block embed all 4 of our animals in the same block. So here we've got a control animal which has just been infused with artificial CSF, and then we've got our animals that were culled at different time points, so 30 minutes, 60 minutes, and 120 minutes, and that's imaged in the same block, as well as some standards which of known concentrations of the Texas Red that we injected into the brain. So this sample block was then imaged on the fluorescence tomography imaging platform, and then the images were captured at 35 micron resolution with the fluorescent tracer, with the fluorescent filter set that we were interested in. So here we were injecting Texas Red conjugated dextran. And so once these images were acquired, they're co-registered to give us anatomical, so that we can see the anatomical bright field images in the same space as the fluorescence images, to get an idea of where our fluorescent tracer is going after we've injected it into the cisterna magna. We then use these image sets for segmentation and analysis, and I'll walk you through some of the analysis that I've done of the data set that we've acquired in a sec. So this was to just highlight the different block sizes that EMIT offers. So in our experiment, we imaged 4 animals at the same time. So we were able to achieve an isotropic resolution of 35 microns. But as Matt already mentioned, the technology allows for different size blocks to be imaged. For example, small blocks where you can image individual organs, or going right up to a larger block which you can image up to 10 vertical mice in. So this is the data that we acquired. So we've got, to orient you, these are 3D maximum intensity projections of our control mouse, our 30, 60, and 120 minute culled animals. So you can see in the control animal, there's a bit of autofluorescence just in the stomach and GI tract, but you can see in our Texas Red injected animals, at 30 minutes we can see that contrast enhancement going down the spinal cord and up into the brain, and you can see the dorsal root ganglion appearing on the spinal cord as well. At 60 minutes, you kind of see quite a different pattern of tracer inflow in the spinal cord compared to the 30 minutes as it becomes homogenized. And then at 120 minutes, you see that the contrast agent goes further down the spinal cord, and we start to see areas accumulating, such as the bladder here. And I've analyzed signal from that data, which I'll show you in a sec. So this was to just show some of the data to you. So I've chosen the 60-minute post-infusion animal. So this is the rotating maximum intensity projection that I've already shown you. And this is just a fly through of the bright field images side by side to the fluorescence images. So you can see that the spinal cord here coming up to the top, and you can see the dorsal root ganglion, which are kind of highlighted either side, and then it goes up into the brain. So by using this high resolution 3D data set, we can then do some segmentation to figure out where the contrast agent has gone and how much of it has accumulated or is currently in each of those regions in the body. So obviously the data that we've got so far using this, studying the glymphatic system, we focused largely on the brain. So that's where I started. So these are just some stills or just some single slices of the brain of our 4 different animals. 20 minutes compared to our controls. And I just wanted to kind of compare it to some of the data that we've got previously. So this is from one of our papers, which we recently published, and this is where we pattern of spread. So at this brain was culled at 30 minutes, and this is that same. This is from the CFT technology, but again 30 minutes. We're seeing the contrast agent go in along the base of the brain as we would expect at this time point with CM infusion of this molecular weight tracer. So quantifying this data, the technology allows, because we've got these really high resolution data sets, we're able to analyze signal in hundreds of different slices. So here what I've done is to just look at all of the different slices which contain the olfactory bulb to get an idea of the mean fluorescence intensity in the olfactory bulb, and you can see that in different levels of the olfactory bulb, right at the end near the eyes compared to where it junctions with the brain, we see quite different extensive tracer. But these are the different groups. So in black here is the control group, and in both the olfactory bulbs and the brainstem, we see increased tracer influx at 30 minutes, which kind of increases again in the olfactory bulbs at 60 minutes before it starts to be cleared at that 120-minute time point. Looking at the cerebrum, so this was the rest of the brain, not including the olfactory bulbs, we see quite a similar picture. So the control animals and then the 30-minute animal here is in pink, as you can see from looking at the nose and at the brainstem here. It's in this orientation. Obviously, we're injecting the tracer at the cisterna magna, which is right at the back of the brain. So we see the tracer going in at 30 minutes, and then we see it increase at 60 minutes, and again, it starts to fall down by 120 minutes. And I've just did some area under the curve analysis of this data, and it shows that kind of signal evolution over time. We're seeing the contrast agent going in, it continues to going in, it then peaks and then starts to be cleared out at these kind of more extended time points. And because all of this data is imaged in the same space, we can then directly compare the extent of contrast agent in each of these brain regions. So what I've done here is just normalize everything to fold changes for control, and you can kind of get an appreciation here of how much tracer is going into these different brain regions over time. So the brainstem is far brighter than the cerebrum, for example, and the olfactory bulbs, and the cerebrum and the olfactory bulbs kind of mirror each other. But the brainstem in this experiment, we were able to see quite a lot more tracer going in compared to the brain regions that we analyzed. So because we've got whole body data, what I then wanted to do was to look at aspects of glymphatic inflow of these tracers which are kind of understudied. So normally we focus all of our attention on the brain. We harvest the brain tissue after we've performed these experiments, but this technology gives us that advantage that we've got the whole body, so we can see where that contrast agent is going. We're infusing the tracer into the cisterna magna, so our assumption is that some of it goes into the brain and then some of it goes down to the spinal cord. But this, from our lab's point of view, is the first time that we've been able to image the whole system in its entirety. So what I did was to kind of compare the acquired data to kind of atlases to get an idea of the contrast infiltration at the different levels of the spine. So by dividing it into the cervical, thoracic, lumbar, sacral, I didn't go as far down past the sacral, but then you can see here by looking at individual slices of the spinal cord as it goes down from the brain down to the tail, you get an idea of how the tracer influx changes over the orientation of the animal. So you can see that here are the different dorsal root ganglia. So you can see we get this spike in signal, which kind of relates to the dorsal root ganglia coming off at the different time points. And then by doing area under the curve analysis of this data, we're able to show quite nicely how the contrast agent evolves over time. So in the more rostral aspect of the spinal cord, we can see that there's more in the at the 30-minute time point, but as you travel down the spinal cord, you can see that the lower aspect of it, we actually see more contrast in the 120 minutes. Again, showing that contrast agent, not only is it being cleared from the brain, but it's also traveling down the spinal cord to lower levels, which we've never really seen at earlier time points, which we generally miss because we take the brains at kind of 30 or 60 minutes. So I kind of dived a little bit closer into this and to look to individual regions of the spine, just looking at those 4 kind of major aspects of the spinal cord. So looking at cervical, thoracic, lumbar, and sacral parts of the brain, and then doing some line profile analysis, so just drawing a line across the section to get an idea of intensity changes and intensity distribution of the tracer in the spinal cord section. And this revealed some quite interesting findings. So in the early time points, in the rostral areas, we see the contrast kind of localized in the the subarachnoid space, and not really much distribution inside the grey matter, as you can see by the pink line here. But as time goes on, at 30 and 60 at 60 and 120 minutes, we start seeing the signal migrating from the outer aspect of the spinal cord into the inner grey matter. We see a similar pattern in the next section of the spinal cord down in the thoracic, and less so in the lumbar. But what's quite interesting is when we look at the cross section of the sacral spinal cord, you can see here that we're seeing much more contrast agent in the grey matter and the inner segments of the white matter in the later time points, indicating that we're seeing this migration of contrast agent going in down the spine and reaching these areas of the central nervous system, which we haven't really studied before. So the last thing I did was to look at drainage and excretion pathways. So once contrast agents are injected into the brain, one of the clearance hypotheses for these is that they get drained to the deep cervical lymph nodes and superficial lymph nodes. And because of the high resolution nature of this data, and we've got corresponding bright field and fluorescence images, what I was able to do is segment out quite nicely the deep cervical lymph nodes and also the superficial lymph nodes to get an idea of the extent of tracer in each of those brain regions. And even though the temporal profile of this data is quite limited, obviously we've only got data at 30, 60, and 120 minutes, but it kind of reveals some clues as to the clearance dynamics of these CSF tracers. At the 30-minute time point, we see the most amount of tracer in the deep cervical lymph nodes, which is kind of in keeping with the literature that people have observed immediate clearance to the deep cervical lymph nodes. But we can see here when we looked at the nasal turbinates and also the superficial lymph nodes, those 2 regions of the body kind of act in tandem, and we see an increase again at 60 minutes, which then starts to die down at 120 minutes, indicating that the tissue is being, the contrast agent, is being cleared out of these regions at the later time points. So to kind of answer where it was going, you might have noticed in the mean intensity projections that I showed you at the beginning. Oh, this is just to highlight that doing some area under the curve analysis, we can get an idea of the different compositions of these regions of the body and how much tracer they're taking in. So the deep cervical lymph nodes seems to be the greatest in terms of how much contrast agent it's taking in. Anyway, so once the fluorescent tracers have been removed or been cleared from these different brain regions, we wanted to see where they were going. So you might have noticed from the mean intensity projections that the bladder starts to light up. So I haven't included the control image, but we see very, very little tracer in the bladder at 30 minutes, but that starts to increase at 60 and 120 minutes, and this signal kind of skyrockets. When we put this in comparison to the other more brain localized excretion routes. We can see that the extent of contrast that we're seeing in the bladder as that contrast is being cleared out of these brain regions is kind of, orders of magnitude higher to what we see in the kind of deep cervical and lymph nodes, and also the nasal terminants. And the area under the curve data there shows that quite nicely. So just to conclude then, I've really kind of enjoyed working with the cryofluorescence tomography, and it appears to be quite an innovative technology for quantifying fluorescence traces in the whole mouse body. From our point of view, it represents quite a powerful tool in terms of assessing glymphatic function, allowing us to look at whole body distribution of traces, which we're infusing into the cisterna magna, which is routine in our field of glymphatic assessment. From the research that I've shown today, we've been able to show that using this technology, we've been able to look at underexplored spinal cord glymphatic inflow dynamics, and crucially, we've been able to see the late stage enhancement within the sacral spinal cord, which to my mind hasn't really been assessed before, and also the temporal migration patterns of how these traces move from the subarachnoid spaces around the spinal cord, and the time profile of how they migrate into the deeper areas of the spine, such as the grey matter. So again, from my point of view, as a glymphatic imaging scientist, this technology represents quite a complementary addition to the kind of suite of imaging, the suite of existing imaging tools that we have for studying the glymphatic system in the mouse brain and in areas of the CNS not confined to inside the skull. So with that, I'd just like to thank Amit for kind of inviting me to give this talk today, my collaborators and funders and members of my lab. Thank you very much.
Thank you so much, Dr. Harrison, for a wonderful presentation. Really appreciate it. Now let's get into the Q&A session. Attendees, as a reminder, please submit any questions you may have into the Q&A panel, and we will address as many as possible. First question, Dr. Harrison, how does CFT compare with your other fluorescence and/or MRI measurements of glymphatic flow?
Yeah, it's a good question. So the good thing about this technology is that we can use the similar mix of fluorescent tracers to what we use already ex vivo. The advantages of this technology, though, is that being able to have to select the tissue that we study. So normally we would do these procedures and we would harvest the brain tissue and just harvest the brain in isolation. But by imaging the whole body gives us the advantage, like we've looked just using this small data set, I've been able to see quite marked changes in the spinal cord. And again, we don't really do that type of imaging with MRI. When we do dynamic contrast enhanced MRI, again, our imaging window is restricted mainly to the brain. So this technology gives us some advantages in being able to look at areas of the brain and areas of the body that we've not really been able to study as yet after cisterna magna infusion of traces.
Great, thank you. Another one here. Have you investigated how flow rate of tracer injection affects the distribution of tracer in the spinal cord at various time intervals?
No, not yet, but that would be a fantastic experiment to do. So what we did, because this was a kind of pilot experiment, what we did was to use the same flow rate procedure that we normally do when we're assessing glymphatic function in the brain. So we just did exactly the same injection experiment. It would be interesting to see that if we kind of increase the flow rate of the contrast agent that we're putting into the brain, do we see more go down the spinal cord? And if it goes down the spinal cord, does it go down there quicker? We've not done those experiments yet, but that would be a fantastic next step to take.
Nico, can I ask an add-on question to that? Sorry to interrupt you. I'm curious, similarly, in doing a sort of MR versus CFT experiment, one might assume that you could then also use a smaller volume. You'd have a concentration difference between a
Yeah,
GAD-based agent and a fluoro-based agent. Is that not
yeah,
true?
yeah, true. So when we're looking at it's all based on kind of the limits of what you can see, right? So with you're right, when we inject the gadolinium-based tracers, even though we can inject them at quite a high concentration, we don't want to go too high because that causes kind of the T1 effects of the contrast agent. We kind of override the signal, which we need it to disperse into the brain before we start to see signal. We kind of get dropout in the back of the brain when we're infusing the signal. And again, when we're taking the whole brain for IVUS imaging, for example, you're kind of based on your the limits of how much tissue or how much contrast agent you can see. With this, we can I've not tried it yet, but I think it would be a good idea to really reduce the concentration that we're injecting and really reducing the volume, because if we can reduce that, we'll kind of limiting the other kind of artifact risks with these types of experiments, kind of increasing ICP and things like that. But
Yeah, we similarly, we're seeing that with a couple other direct brain injection studies where we're having to go back and rethink about concentration and volume,
yeah,
because of the block face gives us a sensitivity advantage.
yeah,
But we're still having to rethink that because a lot of the early experiments, they were MR based. And
yeah,
it's great. It's just a different concentration regime you need to be in, and you're a different sensitivity,
yeah,
effectively. Yeah.
yeah.
Interesting. Sorry, Nico. Thank you.
No, great. Along those lines, another question is, what volumes and concentrations of tracer are you using to avoid detector saturation while also visualizing fine structures like the cervical lymph nodes?
Yeah, so this is I can't remember off the top of my head, but this is the same concentrations of the Texas Red tracer that we've done for brain focused work, and it's in our previous publications. But it's the same concentration of that. If anything, in the CFT technology, we saw so much more signal than what we were expecting to see, and the exposure time was very, very low because we had so much tracer in the brain as it is. So I think if I was to repeat this, I would like you suggest, Matt, is to do much lower concentrations, which would obviously be able to, then we could limit the exposure time, and by ramping up the exposure time then with really low concentrations, we might be able to increase the sensitivity. But right now I've not been able to play around enough to to get more data, but it's planned.
Yeah, Nico, maybe I can add to that too, that in some of the other work, we're looking at like, you know, one or less picomoles
Yeah,
of total fluor that we're injecting, so low concentration. And then I don't think either Ian or I said this, but one of the ways that this system works, the CFT system, is we acquire multiple exposure times. So as Ian just said, like he's using a relatively short, like maybe like 50 millisecond exposure time to analyze his data, but we acquire up to 2.5 seconds. So for distal parts of the body where you might have a lower concentration, we've got the longer exposure times that give you then a much larger uh, dynamic range. This is a unique feature, I think, of the way we do CFT, whereas in nuclear medicine, you wouldn't, I mean, I guess you could acquire a really long time frame and then you could parse out how much data you want to reconstruct at any given, like what part of your list mode you want to use. But one of the nice features here is we do have a bunch of different exposure times to map the space effectively.
Thank you. Dr. Silva, this one's for you. For CFT, is it also used in bioluminescence images or is it only fluorescence?
It's only fluorescence. Yeah, bioluminescence requires living cells and a chemical reaction to take place. So, theoretically, if you had something still glowing and put it into our system with an open filter, sure you'd be able to see it, but CFT is designed for fluorescence. One of the good things about fluorescence as it pertains to sort of durability of signal. So bioluminescence is 20 to 30-minute process to play itself out. Nuclear medicine, those isotopes, very sensitive, but they decay over time. Fluorescence doesn't mind being frozen. So those signals, and we're at very low laser power, so we don't experience bleaching of any of the fluors. So in this case, no, not bioluminescence, fluorescence only.
Thank you. Another one here. Great presentation. Were you able to correlate the line profile analysis with layers of the spinal cord?
That's a very good question. I haven't looked as yet. I'm able to. I've kind of done surface level analysis of this where I've just divided the spinal cord into grey and white matter, and I know that in the earlier time points we're seeing much higher signal in the kind of subarachnoid spaces and in white matter, sorry, and then in later time points we see it into the central grey matter. I haven't done any further segmentation analysis in that, but it's possible because of the high resolution bright field data. If you're able to if you're able to distinguish between the different spinal cord layers on that image, it's certainly possible to then back propagate those ROIs onto the fluorescence data.
Perfect. I think we have time for a couple more here, so we'll keep it going. I think either of you could probably take this one. Since you can see the whole body, is it possible to visualize places like the eye or retina?
Yeah. I don't know if Matt, do you want to go first?
I'm curious what your answer is, but I think you said it right. Yes, you can. I mean, obviously the retina is a relatively thin structure, so you would, we would tend to recommend, as Ian showed the slide, like we can image at a 20 micron pixel size, so you're going to get a few pixels across. I don't remember off the top of my head. I think it's somewhere between 1 to 200 microns in size. So you'd get a few pixels across it. But yeah, we've done eye applications before. We've looked at optic nerve or trigeminal nerve outshoots around the eyes. So you certainly can. But what was your perspective? Did you also take a look at the eye?
Yeah, I was going to say when I did a little bit of segmentation looking at the optic nerves and trigeminals, like you say. I didn't actually segment out the eyes to see if they've got signal. I don't know if they would have signal after you infuse a tracer like this, but people have done kind of glymphatic measurements and looked at optic glymphatic function in the optic nerves. So with this technique, it definitely be possible. I guess, like you say, Matt, it would probably be a lot easier to go with a higher lower pixel size to get kind of increased resolution, because the structures are a lot smaller than the kind of whole spinal cord images that I was looking at. But yeah, definitely possible.
Yeah, the other advantage there, Ian, is that in order to get higher resolution or smaller pixel sizes, we move the detector closer to the sample, so you also get additional sensitivity that comes along for the ride.
Yeah.
The other thing I'd point out, actually, a lot of the eye work that we've done actually has been in larger animals. So, for example, a non-human primate, where you can just run the eye in isolation. So we can just do that part rather than, unless you need to see the whole sample. But if your question is eye only, that, running just that tissue alone is possible.
Dr. Silva, a question we get often. Is it possible to recover any of the tissue slices?
There's a couple of ways that we do that. Number one, so not slices, but we will do tissue biopsy, the same as you would like for dermatology. And it's frozen tissue. So anything that you can do with other frozen tissue, you can do a post fixation if you want, or you could take it to do PCR, or if Ian wants to do ICP-MS or something to look at the gadolinium. If he did a co-injection, he could do that with the tissue. Or you can collect tissue onto tape and transfer it to glass slide to do work on glass. So those are the two methods for collecting tissue.
Great. We have a little bit more time, so let's get these last two in. Dr. Harrison, have you tried different forms of anesthesia, and if so, does it affect distribution or rate of clearance?
Again, no, I haven't, but we've done kind of anesthesia experiments just looking at the brain in our other work that we've done in the lab. But using this technology in the whole body setting, we've not looked at different anesthesias yet. So I don't know if we switch to, if we go to isoflurane or ketamine and xylazine, we know that those two different anesthetics are quite they differentially affect the glymphatic system. I don't know how they would affect the ratio of brain to spinal cord inflow. So that'd be quite an interesting question to ask. So yeah.
Okay, great. Last one, Dr. Silva, do you use a particular diet to minimize autofluorescence in the GI tract?
That's a great question. The short answer is yes. The longer answer is that it's really dependent on which channels are going to be included in the study. So at this point, I think we've looked at about 30 to 32 different animal channels. We've characterized them in all of the channels that we have. And so then we can provide some guidance when starting an experiment to what food to use. The second part of that, though, is that it really turns out to be more nuisance than complication. So we'd rather not see really bright guts, but it doesn't, in the case of this study, we saw some signal, I think, in the stomach with Ian's animals. It doesn't impact the ability, as long as you know what that is, it doesn't impact the ability to see the brain and the cord and some of those other regions. So it's something that we do think about and are looking at, multispectral approaches or unmixing approaches that will allow us to get rid of it as well. Just more complicated math in the context of a three-dimensional, large three-dimensional study. So yeah, thank you.
Great, thank you. That's all the time we have for any questions. Thank you, Dr. Harrison, so much for joining us today. Thank you, Dr. Silva, and especially to the attendees and all the great questions. Really appreciate it.
Bye, everyone. Thank you.